So our next speaker is Patricia Tran from here at the University of Wisconsin.
Hello, can you hear me? Okay. Hi, everyone. First off, I'd like to thank the organizers and everybody involved for inviting me to present today. My name is Patricia, and I lead the Bioinformatics Research Support Service here at the University of Wisconsin-Madison in the Department of Bacteriology. And I obtained my PhD in the interdisciplinary field of environmental sciences and microbiology. And I did a lot of field work, laboratory work, and computational work to try to understand what microbes are doing in environments, specifically in anoxic environments. And today in my current role, what I do is mainly to help graduate students, postdocs, and faculty use the computational resources that are available on campus so that they can actually do the research that they want. So today I'll be speaking about what it's like to support microbiologists in research computing. So big data is really common in biology. For example, researchers are routinely generating billions of DNA sequences that contain the genetic code for all life forms, from bacteria to plants to trees. And I mean, trees are plants, I'm sorry, to all kinds of life forms. And the sequencing data is deposited onto databases such as the NCBI GenBank that contains over 3 billion sequences representing over 500,000 species. In another subfield of biology, we can think of proteomics, which is the study of proteins and their functions. And these data sets have been used in models such as AlphaFold 3 to be able to accurately predict how proteins will fold. But the data that goes into these really large-scale models has to be made or characterized in the lab. So, for example, the protein data bank contains over 210,000 experimentally derived structures by researchers. But it doesn't necessarily mean that it's only in these big databases. Researchers on the day-to-day in the lab are doing experiments at the scale of hundreds of samples, as demonstrated here by these 96 well plates, where each of these individual wells represent a culture of bacterium that's associated with its own data. Now, these different types of data forms are used to answer all kinds of questions in biology, from what is the function of a gene in a metabolic pathway to what is evolution looking like for a certain function of interest, and also to answer questions about microbiology and disease. So as you can imagine, these data forms in biology comes in all data types. It can come from different sources in quantities that range from something you can analyze on your laptop to something that is very impractical to do on a laptop. And like in many fields of science, there is a demand and need for that to be reproducible, especially when it comes to computational biology. And like in many fields of sciences, there's also collaboration that happens either between lab members or labs in a department, institutions, and even countries. And these data sets have actually been more prevalent over time. On the figure on the left of this slide is showing the cost of generating sequencing data over time. And maybe 25 years ago, it cost over $10 million to sequence one raw megabase pair of DNA, and then advances in sequencing technology around 2007 have really dropped this cost and made this a lot more available and routine across all labs. So today we can sequence something for like $10 a sample. So a lot of these labs across the field of biology are generating data, depositing that into public databases. This is data from the NIH showing about 36 petabytes of data in the sequence read archive. Now we think that the rise in data generation and availability in many fields of biology requires scalable, flexible, and high throughput computing. And this is where we think domain-specific research support can be really helpful. So before we started our research support program 2 years ago, the main ways that researchers were getting help about research computing and bioinformatics sometimes was very local. And this is a situation that a lot of labs across the country are encountering, where there's a lab, there's a graduate student, a very motivated postdoc who knows bioinformatics, and he or she is teaching that to the lab members, but obviously they will move on to another position. And the knowledge about this critical piece of the biology workflow is lost along the way. And so the type of research support that these different labs is really variable in the field. And on the other hand, I want to give a shout out to the campus level research facilitators who have a really broad reach of fields. So they can help out many people in different fields of sciences in research computing, but it is out of scope to try to teach the researcher, okay, what are the best bioinformatics software, like what are the parameters to use for their specific research questions, just as an example. And so overall, our mission is to help microbiologists of different career stages, but also knowledge backgrounds, to make the most of the computing resources that are available to them so that they can succeed both professionally and also personally. So in order to do that, we focus on the whole life cycle of a project. And even if today I'm mostly going to be talking about the analysis part and working with the Center for High Throughput Computing on supporting microbiology related computing, we also support researchers during the planning phase, the experimental design phase, and also what happens to the data and the analysis once the project is completed. And this is a really helpful model for us because we can anticipate what the computing challenges are going to be by listening to the researchers and what their research motivations are before they get there. And so we can think of how we can design workflows that work for them. And like, if they want to learn something really complex about scaling up their analyses in 5 months, what are some of the things that we can prepare them to, in these next couple of months so that they can do that then? So we do serve a variety of people with different majors or concentrations, but there are some common challenges from the learners that we hear a lot. The first one is related to new vocabulary related to computing. So keep in mind that these are experts in their own field. They are biologists or microbiologists or physicists, and they have a certain expertise. But learning about computing can be really overwhelming because it's like being an expert in 2 things. So we help them with that process. Another challenge that we hear often is that there are a lot of materials or documentations or resources out there, but there is a gap between how do we use that material and implement it in their own work. And obviously in scientific research, every research question is going to be different. We're trying to answer novel research questions. And so the balance between making documentation and resources that are general enough so that many people can use versus how do we actually implement that to answer a very specific question. So in the case of high throughput computing, when we talk to the researchers and we figure out that this is an appropriate computational framework for them to use in their research, there are some common reasons that these people are coming to for help. For example, of course, there are HT Condor basics, how to log into the systems, access the resources, submit files, look at jobs, fix typos, and then also using containers. Here also, we don't have domain-specific software installed on all the working nodes. And so the students or the researchers have to learn how to use softwares, how to use containers. And you can imagine that's a pretty big learning curve. A lot of the steps in bioinformatics require, a lot of analyses in bioinformatics require multiple steps. And so how do we automate these complex workflows so that the person doesn't have to wait for their job to run before going on to the next step? Turning local scripts into scripts that are suitable for high throughput computing. Many people come in with experience maybe working on the terminal on their laptop, but to use that on a high throughput computing systems, then we need to learn how to make these scripts reproducible, and then that can just be run on a working node. Troubleshooting infrastructure issues. I think anybody who works in coding has worked on troubleshooting code, but that's sometimes an assumed skill from people, and people actually like to learn how we troubleshoot, seeing examples live of troubleshooting, and that's really helpful for them. And of course, domain-specific analyses and interpretation. So recommendations on what are the best practices in the field, what are different softwares doing, how does that intersect with the data that they're having. So how do we take someone from a high throughput computing novice to someone who is independent, confident, and have the results that they need so that they can move forward? And I intentionally did not use the word expert here because it really depends on what the users are needing. So to demonstrate this point, I'd like to highlight the work of one of our researchers in our department, who is Dr. Charlie Moe. And he works on bacteriophages. And about a year and a half ago, he sought our research support program to learn about high throughput computing and how they can use that to analyze the vast amount of data that they were generating in the lab. So in this project, we looked at methicillin-resistant Staphylococcus aureus. And Staphylococcus aureus is a type of germ that's present in about 30% of the population. And most of the time, it's really harmless. It doesn't cause anything, but in a small fraction of cases, it can lead to infection and really severe consequences, including death. So what are people doing? They're trying to apply antibiotics, but sometimes these antibiotics are not working. And that's the case of these methicillin-resistant Staphylococcus aureus. And the observation that sparked their project was that when they took a MRSA strain, this antibiotic resistance bacteria, and they evolved them with the phages, they saw that in the evolved versions of the phages, it actually modified the behavior of these bacteria and their resistance to antibiotic. So what you can see on this panel C here is 3 MRSA strains, the parental strain and the evolved strain. The beige background is a colony of bacteria, and this white strip in the middle is an antibiotic strip. And what you can see is in the parental strain, the bacteria is still growing, so it's resistant. And in the evolved strain, the one that was subject to the bacteriophage, it had a loss of antibiotic resistance. So this sparked a number of experiments related to genomics, experimental mechanistic lab work, and transcriptomics to try to understand that. So we used high throughput computing, and we taught the users how to use HT Condor, how to automate jobs, but also we wanted to relate these high throughput computing skills to what they were doing in the lab. So how does the experimental design going to affect the amount of data that they're using and then analyzing on this system? We also had to work with various providers. In microbiology, it's very common that part of it will be done in the wet lab. We'll maybe ship some samples to another center to get some sequencing data back, receive it in all kinds of formats depending on the center. And then we have to bring that data onto the high throughput computing system, run it at scale, and transfer that data back. Also, we had to focus on skills related to critical thinking. I don't know if there's any experimentalists in the room or have experience running an experiment by reading a methods section of a paper, but it's really something to turn a paragraph into something you can do in the lab. And it's similar with computation. So how do we turn a paragraph into a series of code that we can run? And then finally, it's also useful to use high throughput computing because we can make sense of the logs, the submit file, the executable files that don't change. And we can use those to communicate the science after by writing papers. So specifically for this project, there were some tips and tricks that we implemented in HT Condor. For example, we had a passage experiment that had multiple variables from bacterial strains to treatments to a time element. And we incorporated that into the metadata spreadsheet that we use in a queue statement of a HT Condor file. And we made that flexible enough so that as the project evolves, the researchers can use the same template depending on their experimental design. And we also had to deal with various lines of evidence, which means various workflows, various softwares, and so on. And so we use Dagman to try to connect all these pieces together. And we also created container images so that this was usable by these researchers. And something I want to highlight here is that the skills that we focus on teaching the researchers really depends on their learning goals. Some might be really interested in learning about the behind the scenes, but some are just okay learning the basics, and that's okay because we want to keep the researchers interested and motivated to learn as they progress in their research. So finally, I just want to end with some lessons learned from what it's like to support research computing for microbiologists. The first one is that there is an impact way beyond the technical skills. What I hear a lot from students is that they often feel really appreciative to have a go-to person that they can meet with and ask their research questions and meet them where they are in their learning process. And so they often report that they can feel less afraid of bioinformatics, but they can also see how high throughput computing complements their major area of research. The second thing is that the research needs and questions are going to naturally evolve over time over the time that a project happens. So as research facilitators, I think something that we can do is to create space and opportunities for frequent interactions with the researchers so that they can feel comfortable to come and ask us questions. And in fact, 60% of the people come to us more than once. And that's expected because they can't learn everything in one session, of course. The third one is that each user is unique, but there are some common trends in terms of learner profiles. So knowing this can guide outreach efforts. So for example, in the past 2 years, we were able to guest lecture in graduate level bioinformatics courses. We started a campus level container registry for bioinformatics softwares and started making video documentation, not just on HT Condor skills, but with examples from biology. And then finally, I think this was mentioned in some previous talks today, but developing rapport and trust with the labs and campus partners is really essential. So in the past year, we started to meet monthly with the CHTC research facilitation team, and we can tell them directly about what microbiologists are using high throughput computing for. And they tell us about what are the new latest features, and I can communicate that with our immediate research community. So having these interactions and is helpful to receive feedback and inform our future strategy. So on that, this is where I'll end my presentation. Thank you so much for your time.
So we probably do have time for a question or two while we're switching this.
So, sort of a comment. I am not aware of a feedback channel from you. I would say to us, I was wondering whether there are opportunities for you to, in the name of translational research, to tell us more about what you find missing or what you find that we can do differently. I understand that you are meeting with Christina once a month. I feel that it's not necessarily frequent enough. And I would like to make sure that you are at the front. And I also wonder whether there are a topic or problems that you are facing that you want to escalate to us as a tier 2 support, because we have the technical expertise that you don't have and you should not have, right? So I don't know if you have immediate thoughts on this. If not, we should have a further discussion.
Yeah, totally. Just to give out one idea, something I think that's maybe biology like related is like a lot of our bioinformatics workflow, even if we break them down into like running per sample or different steps in the bioinformatics workflow, they're usually like extremely memory and intensive and disk intensive. So even though high throughput computing works well most of the time, there is still quite a time to wait for jobs to run or like for things to test, even when we scale that down. So I guess like that would be one thing on my wish list of something to discuss.
So this came up yesterday as well, that and I think also in Eric's presentation about his workflow, that there is a node. Now, if we better understand these requirements, then we may do better, but we need to go back and forth also to impact our investment in capacity and whether we do something in our scheduling to address, but you understand, we have to go back and forth and not just assume that it's all a cookie cutter.
Totally. Thanks.
Thank you, Patricia.